Hi everyone. Today's topic of discussion is provided by visual processing, by vision. Now, there are quite a few senses that we could discuss with minimal detail, but instead of doing that, we're gonna rely on a tradition for introductory courses and instead spend a bit more time and focus on vision. So again, our brain provides extensive resources for the processing of a number of different sensory modalities. Vision, auditory perception, both of which are quite well described in the textbook, along with tactile perception, smell, and so on and so forth. However, we focused on vision, at least for the purpose of the lecture, for two main reasons. One is that the brain tends to allocate a substantially larger amount of resources to it, as opposed to other senses. And secondly, because there is an extensive, a vast body of research conducted for many decades across the entire world dedicated to vision. So in sight of that, also vision research is quite well represented at the UTSC on our campus. We have quite a few members of our faculty, including myself, who dedicate much of their research careers studying a variety of phenomena in vision. So without further ado, today's discussion, it is about vision, and we're going to start with trying to understand how external stimulation, in this case light, reaches the eye. It is transduced into a neural signal, and how this signal reaches the cortex, the occipital cortex. Next, we will proceed with a brief discussion of certain cortical areas engaged in color and motion perception. Then we'll see how information is processed across a hierarchy of visual regions within the temporal and parietal lobes, achieving higher and higher levels of complexity while also acquiring more meaning and also more functionality, more practicality, for instance, in guiding movement. Next, we'll have the opportunity to examine some bewildering phenomenon called synesthesia, which has captured the attention of researchers and of the public for quite a few years, where different senses, including vision, start to crosstalk, leading to very interesting phenomenological results. And last but not least, we'll try to bring to a conclusion our conversation from last week on how to proceed with conceptual and operational definitions guiding the study of specific cognitive processes. Okay. So one distinction that you might be familiar with from introductory courses in psychology is that between sensation and perception. And this is an old distinction, perhaps even somewhat outdated. Still, it is quite useful to emphasize a critical distinction. That between the transduction, the conversion of an external stimulus, such as light in the case of vision, or pressure wave, air pressure waves in the case of audition, into a neural signal. This is a very interesting biophysical process, and we can probably dedicate an entire lecture, if not an entire course, just to its study. It is truly fascinating how a signal from one modality can be converted into a signal that a biological system, our eyes and our brains, can process and make sense of. And now that we talk about sense, the other aspect of this distinction is provided by what we call perception. Sensation roughly covers the conversion from external stimulus into neural signal. So then the other side of the distinction covers the ability to process all these sensory signals, integrating them and trying to make sense of them. Now, in the case of vision, what happens is that light falls on the back of the retina, on the back of the eye, and then all the signals processed over there by an entire patch of neurons, photoreceptive cells, provide the sensory input for the brain, which tries to make sense of that extensive and highly complex sensory input through multiple layers and levels of neural processing. Okay, so this is the distinction between sensation and perception. So before we move into perception, let's take a quick look at how sensation occurs in the case of vision. And it's all got to do with the eye, with the anatomy of the eye and the functionality of the eye. As you already know, light passes through the lens. It is refracted through the lens, and it reaches the back of the eye, a densely populated patch of layer of neurons, of photoreceptive neurons. So what happens here? And again, we can go into too much detail, but we should know the very basics of the anatomy of sensation in this case. Well, what happens is that we have two populations of cells on the retina. One is provided by rods and the other one is provided by cones. Cones are color sensitive and they densely populate this middle central part of the retina, which is called fovea. So whenever you're looking at something on the screen, let's say that you're fixating on this particular spot on the screen while watching the lecture, then what happens is that essentially you're projecting the visual information corresponding to that patch, to that location on your screen, to the back on the eye on your fovea. So you stimulate a maximum number of cones to that information. Now, cones are not extremely sensitive to the light, which means that they take a higher amount of light to be brought into action to adjust their firing rates. However, because of the extensive, the amount of density of cones on the fovea, that means that whatever you fixate, whatever you look at, is processed with a high degree of visual detail. Fovea and the cones that populate it provide you with high level of visual acuity. In contrast, whatever falls in the periphery, so let's say while you're focusing on this particular part of the screen, then other parts of the screen such as eye here, the word eye or the word ganglion cells here, fall on the peripheral part of the retina. And now the peripheral part of the retina is much more densely populated with rods. Now, rods do not have the color sensitivity of cones. They also do not provide the same level of visual acuity, but they take a lot less visual stimulation, light stimulation, to respond, which makes them exceptionally useful in the evening, at night, or within poor lighting conditions. They allow you to sense motion within the periphery, and accordingly, they allow you to take action, right? So rods and cones have complementary roles in facilitating high acuity central vision during normal or good lighting conditions, as opposed to higher light sensitivity within the periphery of your visual field within poor lighting conditions. Okay, so this is what happens on the retina. Light falls on the back of the eye. Through the process of transduction, the light is converted into action potentials, firing rates. So then what happens next? Well, the signals yielded by the rods and the cones are next passed through another layer of neurons, which are labeled here as the middle layer, and then the ganglion cells. Next, what ganglion cells do is get a signal, and then through a bundle of axons, they pass this signal through a gap within the retina. So all this bundle of ganglion cell axons provide the optic nerve, which leaves the eye and then connects to the brain. Okay, so you can think of it as the cable that connects the eye to the brain. As a result of this arrangement, of this architecture, some very interesting phenomenon occurs. And that is the result of having this substantial gap within the back of your eye. So that means that whatever information is passed through the lens to the back of the eye is actually not properly processed. There is no patch of neurons, photoreceptive cells, either rods or cones, that are responsible for processing that information and carrying it to the brain. Yet, we don't need to see it. In normal circumstances, at least, we do not seem to suffer from that gap in sensory processing, nor are we even aware of it. And that happens because we have the amazing ability to make up information. So essentially, whatever this eye cannot process, the other eye might be able to. So the other eye can compensate for the lack of information that should occur here. We also appear to integrate information. So whatever happens in the proximity of this gap, of this blind spot, is the result of trying to infer based on what happens in the proximity of this gap on the back of the retina. So the eyes and the brain have an amazing capability not only to process available information, but also to infer what is missing, and hence to convince us of the reality of what's in front of us, but also of what is actually a piece of fiction. Now, there is quite a few number of interesting demos in psychology that allow you to examine the strange reality of having a blind spot so close to your central vision, so close to the fovea. So you're welcome to try any of those if you haven't already. Okay, now I have mentioned that rods have amazing sensitivity to light. And again, this belongs to the study of sensation, so we don't have a lot of time to discuss this in particular, given our focus on perception. However, I do want to bring you to your attention something that became quite exciting to find out about over the last few years. So it's been known for a while that rods have the ability to adjust their firing rates in response to a single photon. So for those of you who do not remember, a photon in the study of quantum physics is the smallest unit of electromagnetic radiation, right? So photons are extensively studied in quantum mechanics, for instance. And it's been known that our biology is extraordinary. We have rods that can capture and become sensitive and yield action potentials accordingly in response to a single photon. What has been proven more recently, though, is not only that our biology is sensitive to that, but our psychology is sensitive to that as well. So in psychophysical studies, it's been shown that humans are able to detect above chance the occurrence of a single photon falling on our retinas. And this is quite extraordinary because for many people, this essentially builds a bridge between quantum mechanics, neuroscience, and psychology. So currently this has yielded some very interesting speculation on how to bridge the study of very different disciplines. Okay, unfortunately, in many cases, our sensitivity to light information is compromised and even entirely lost. Sometimes that happens because of how our brains fail to process visual information, but in a whole number of cases, they actually have to do with the biology of the eye, the physiology of the eye. Perhaps many of you are already familiar with terms such as detached retina, where a whole number of layers of photoreceptive cells are displaced, or with macular degeneration, a degeneration of the layers containing photoreceptive cells. So in both of those circumstances, the problems can range from mild to dramatic. There is a loss of vision. And engineers have studied for many years the ability to recapture, to regain visual abilities to at least some extent. And one of the devices that has been particularly paid close attention to is the retinal implant in its many different variations. And one of such, actually two of such variety of retinal implants are shown here. One that attempts to replace essentially non-functional photoreceptors and pass the information directly to ganglion cells. And the other one that tries to enhance still functional but diminished photoreceptive cells on the back of the retina. The textbook describes in a bit of detail what happens with the epiretinal implant. So this communicates with a small external camera via Wi-Fi signal. And then via small electrodes within the implant capture light information that would in normal circumstances be distributed across hundreds if not thousands of photoreceptive cells. So a result of that, some sensitivity to light and to motion, and even the ability to vaguely process the contours of objects is recovered. However, the resolution of vision afforded by a retinal implant is nowhere close to the one that we currently have in virtue of our biology. So plenty of work remains to be done in this domain, and alternatives to retinal implants relying on optogenetics are also currently being explored extensively. Now, some of you have asked how important are diagrams within these lectures and within the textbook for understanding the course material. And I would say that for most of them, a fine level of detail being processed is not really necessary. Keeping track of all the terms and the geometry of the diagrams and the figures, well, this is an exception. This diagram is essential to the study of vision. So let's delve into it, into some detail, because it is also the source of quite a few common confusions for students who take introductory courses in psychology and neuroscience. Okay, so what this figure shows us is how information within your environment is processed and passed through the visual system to reach the occipital lobe, to be specific, the primary visual cortex area of the occipital lobe. Okay, so what happens here? First of all, we have to become acquainted with some terms. one of the key terms here is visual field, and that is essentially what is in front of you. So whenever you're looking, let's say at the center of your screen right now, whatever's on the left hand side of it is labeled as the left visual field, and whatever's on the right is the right visual field. So that way we can have a map of what's right in front of us and being refracted on the back of the retinas. Now, this process of refraction, just in virtue of simple geometry, yields some very interesting constraints on how information is processed. A common source of confusion, or rather a wrong belief, is the fact that each eye processes only one visual field. Well, that is wrong. That is a mistake. Each eye processes, to some extent, each visual field. So, for instance, if we're looking at the left eye here, we see that the retina can broadly be described as made up of a nasal part and a temporal part, and we call this nasal because this part of the retina is closer to the nose, right? So then, in virtue of geometry, a part of the left visual field falls on the nasal side of the retina of the left eye, while the right visual field tends to be projected on the temporal side of the retina of the left eye. And then the converse result occurs if we look at the right eye. So then the temporal retina of the right eye captures the left visual field, and the nasal side of the retina of the right eye captures the right visual field. Is there a complete redundancy in how information is represented? Well, not exactly, because again, just in virtue of geometry, the nasal side of the right eye might be able to capture something which is falls on the extreme periphery of the right visual field, while the temporal side of the left eye only has the ability to capture that much of the right visual field. So it's got some restrictions in that sense. So there is a fair degree of redundancy in parts of the visual fields that are processed by both eyes, but there are also some relative advantages and disadvantages, again, in virtue of the geometry of light refraction. Okay, so this is the first stage of processing. Light falling on the back of the eye, being processed by the nasal and temporal sides of the retina. What happens next? Well, interestingly enough, the nasal side of the retina of the left eye and the nasal side of the retina on the right side cross paths, so the axons that carry information via the optic nerve from these parts of the retinas of the two eyes, cross paths at a location called the optic chiasm. And as a result of that, whatever represents the right visual field on the back of both eyes is projected on the right side of the thalamus to an area called the lateral geniculate nucleus, or LGN for short. This is the first stage of visual processing within the brain. Conversely, the information which corresponds to the left visual field is projected to the right LGN within the right side of the thalamus. Then the information from the two LGNs is radiated, is conveyed, it is passed to the primary visual cortex of each hemisphere via the optic radiation. So we have the optic nerve passing information from the eye to the LGN, and then we have the optic radiation passing information from the LGN to the primary visual cortex. And we call this the primary visual cortex because this is where information from the eye via the LGN first lands on on the cerebral cortex. Okay, so then in virtue of this arrangement and in virtue of the anatomy that underlies it, brain damage to various cortical and subcortical areas can have some very clear-cut and obvious consequences. So for instance, because the right primary visual cortex is responsible for the left visual field, if you damage it, then you will no longer be able to process what happens on your left side. And this happens despite the fact that eye function can be entirely healthy, normal. It can also happen despite the fact that the LGN and the thalamus, it does its job. It's just that information no longer reaches the primary visual cortex. Hence, a critical step in visual processing corresponding to left visual field information is missing. And then converse of this happens if you damage a right primary visual cortex. As a result of that, you will no longer be able to process information from the right visual field. This is cortical blindness. We no longer are able to perceive what happens on within one visual field or the other. Similarly, if you damage the right LGN, you no longer process what's on the left, and if you process the left LGN, you are no longer going to be able to process what's on the right. So then one might wonder, what happens if you damage the optic chiasm? And I'm gonna invite you to pause the lecture for a second and think about it and see if you can come up with the right answer. Well, assuming you have done that, then here's what happens. I believe I already mentioned the fact that while some redundancy occurs in terms of what the eyes process as visual information, there is a certain advantage. So for instance, the nasal side of the right eye might have access to more peripheral information than its temporal counterpart on the other eye. And the converse happens with respect to the other visual field. So if you damage the optic chiasm, then you're losing information with respect to some of the extreme periphery of the visual field. The outcome being that you will be limited to tunnel vision. So you're still very good at processing what's right in front of you, but your ability to process visual information within your periphery will be impaired. And one last thing that I should mention is what happens if you damage one eye versus the others. So again, if you damage one eye, that doesn't mean that you're completely limiting or eliminating the ability to process one visual field, but rather you have some limitation to processing information in both visual fields. Okay, so we know this from decades and decades of studying physiology and performing a variety of experiments, both in the human visual system and the other primates, and also from neuropsychology, right? So lots of studies of brain damage inform and confirm this understanding of the architecture of the visual system. Now, we have seen that in virtue of our anatomy, we all have a blind spot, right? It is caused by the absence of a appropriately placed patch of photoreceptor cells at the location where the optic nerve leaves the eye and the retina. However, additional blind spots can occur because of brain injury. And to have a better understanding of that, we'll have to reintroduce the idea of retinotopic mapping on primary visual cortex. And this relates to a discussion that we had in the previous weeks, more generally, not just about vision, but also about other senses. To be more specific, we saw, for instance, that somatosensory cortex maps very precisely different body parts, right? We saw that auditory cortex maps quite well different frequencies associated with sound. And we also saw that the primary visual cortex maps areas of the space, areas of the visual field. But to be more precise, because now we can, what primary visual cortex maps exactly are different parts of the retina. And because different parts of the retina correspond to different parts of the visual field, which project on them, this is how indirectly the primary visual cortex ends up representing with different parts, different segments, different areas of the visual field. And this bullseye display hopefully conveys that point quite well. What this shows is that, and we already know the first part, one half of the visual field, let's say the left half of the visual field, is mapped onto the opposite occipital cortex, onto the right occipital cortex, and vice versa. The other thing that is apparent here is that not only are things flipped left right when it comes to visual field mapping onto the visual cortex, but also bottom up. And what I mean by that is the upper part of the visual field is mapped onto the lower part of the visual cortex. So to take just one such example, if you're looking at the top left visual field containing these areas 1, 5, 9, you can see that they are mapped onto the lower part of the right visual cortex. So we have 1, 5, and 9. And this leads to some very consistent and systematic decrements in vision in the presence of specific type of injuries, as introduced, for instance, by stroke. So if you damage a relatively well delimited and relatively well spatially constrained part of primary visual cortex, then you are indirectly affecting the ability of someone to process the corresponding part of the visual field. And we call this a scotoma. This is essentially a blind spot caused by a localized brain injury within primary visual cortex. And sometimes when you look at these patients, you can see things like that. So you can see here, for instance, in polar coordinates, a map of the visual field for the corresponding to the left eye and the right eye. And then we see below a sagittal section through an MRI scan of the patient. And this is quite typical of what happens when somebody goes through a brain injury of primary visual cortex. So now, just by examining this image, where the bright white spot indicates the location of the injury, you might be able to infer what part of the visual field is no longer available to the patient. So you can see already that this is pretty low, right? So it's affecting a bit more the bottom part of visual cortex, which means that it should be corresponding to the higher part of the visual, to the top visual field. And presumably this has impacted the left hemisphere, though you might not be able to tell from this picture, but you can infer that by looking at the part of the visual field that is displayed here in black on this map. So typically there's a very good correspondence, and one that you can predict just by examining either the injury or the part of the visual field that a individual no longer has access to. Does that mean that a patient is fully aware of the presence of the blind spot? In many cases, where the damage is relatively well spatially constrained, that's not the case. Just like the physiologically normal blind spot is covered up essentially by our visual system through a process of reconstructing, or rather inferring, what might be happening in that part of the visual field through a process of inference, through a process of interpolation. The same can happen to blind spots caused by brain injury. So you might have a scotoma, but it might take a while to realize that this is actually something that is not going well, right? Things might simply just disappear from your field of vision, but that doesn't necessarily mean that you will have a very clear-cut absence of information that you can become aware of in that particular area of the visual field. Okay, so this is what happens if that injury is relatively well delimited. But what happens if the entire visual cortex, if the entire primary visual cortex becomes dysfunctional or damaged? Well, then we experience cortical blindness. And we say blindness because the eyes might be functioning just perfectly, the LGN just as well, and so on and so forth. But what happens is that we exhibit an inability to process visual information. So in that sense, we are blind despite the fact that everything else that precedes in terms of visual information processing, the arrival of that information to the primary visual cortex is still fine. Okay, so this is cortical blindness. Does that mean that you exhibit a complete inability to act on visual information because the occipital cortex no longer functions? Not exactly. So as I mentioned earlier, probably the LGN is still doing its job, and so are other subcortical structures that are fed with this type of information. So you might be able to act on visual information even if you're not aware of it. And those are some very interesting phenomena that unfortunately we're not going to have a chance to talk about at length in this course. So for now, this is what we need to remember. There is a type of extensive brain injury that leads to cortical blindness, inability to process visual information. And this is something that patients many times manage to recover either completely or partially from, but naturally sometimes that is not the case. Okay, so then let's move on to a related question, and this has led over the last decade and a half to some fascinating research. It relates to brain remapping and brain reorganization in the presence of congenital blindness. And this time we're talking about actual blindness, the result of eye dysfunction, inability to process light and light information because the eyes are not doing their job. Well, what happens in this case is that many of these individuals experience a remapping of occipital cortex. So we already know that the occipital cortex is responsible for vision. So what happens if from a very early age the occipital cortex is no longer receiving the visual information that it does in most healthy individuals? Well, you might imagine that the brain is quite pragmatic and opportunistic in that sense. So then it uses the neural resources housed by occipital cortex to do other things which are quite important in these individuals. And one of the things that it does is mapping sensory information necessary for Braille reading. So here we've got some fMRI pictures projected on the surface of the skull, and we can see here how while reading Braille, a lot of these individuals exhibit entire areas of occipital cortex being activated, coming online. But the story gets even more fascinating because, at least in some individuals, not only is the occipital cortex recruited for Braille reading, but it also brings into play a very different type of mapping, akin to what we see in somatosensory cortex. To be more precise, what happens is that we get a mapping of fingers onto different parts of the occipital cortex. So clearly the occipital cortex, and the primary visual cortex in particular, is prone to a systematic way of mapping information spatially. So in this sense, one could say that perhaps the occipital cortex is not necessarily for vision per se, as it is, for instance, for object recognition, right? Recognizing letters, words, and sentences. Okay, so this is a truly fascinating and relatively new area of research, and to some extent what happens to the occipital cortex can also happen to other areas of the brain, such as the primary auditory cortex in people who are deaf. Now, as an important caveat, if blindness occurs later in life, it is very rare to see this type of massive reorganization. So again, brain plasticity is primarily seen most extensively and clearly at an early age, and even more so when deprivation of sensory stimulation occurs from birth. Now, a lot is known about the occipital cortex and the primary visual cortex, especially area V1. But then it is probably time to move on and see what happens beyond area V1. Well, V1 is neighbored by an entire series of cortical regions with names such as V2 and V3 and V4, and each of them have very interesting and specific functions. They build on the information that V1 provides to acquire more sophisticated models and representations of the visual world, and to feed those into a pathway dedicated for visual recognition and to one dedicated for action. More about later. However, since we don't have time to go into the details of every single one of these regions, let's pay a bit of attention to area V4. And the reason for that is both because of its unique significance and because of its history. In the 70s, Semir Zeki, a British neuroscientist, conducted a lot of research in monkeys, and his neurophysiological investigations led him to conclude that this specific area of visual cortex in monkeys, which he called V4, seems to be particularly sensitive to color processing. So that doesn't mean that it is the only part of the brain sensitive to color. By no means. V4 receives information from other cortical areas, and V1 in particular. So color information is already passed through V1 and V2 and so on and so forth. Also, it is not the case that V4 only processes color. It also has sensitivity to some simple geometrical shapes. But by and large, V4 exhibits considerably more sensitivity to color than what we might expect from other cortical areas. So now this was in the 70s and relied on monkey research. So with the advent of neuroimaging in the 90s, Zeki was quite curious to identify the possibility of V4 in humans. And at the time, PET was particularly popular for neuroimaging purposes. fMRI became a much more available tool at the end of the 90s. So in the early 90s, Zeki looked at the ability to discern areas sensitive to color using PET. And actually, he proceeded in two steps. In the first step, he looked at what happens within the visual cortex when an individual has the eyes open versus closed. And what he found, as expected, was that the occipital cortex was a lot more active in response to the first condition relative to the second condition. There was a lot more blood flow to the occipital cortex when an individual kept the eyes open rather than closed. And as we already know, blood is needed to support the metabolic cost of neural activity. Okay, but this is just a proof of concept that PET is delivering a meaningful signal and nothing else, because otherwise we already know that the occipital cortex is dedicated to vision, and vision implies many things, including but by no means limited to color, right? So then what Zeki did, and this is the critical part of the experiment, was contrast using the subtraction method a condition in which a participant saw geometric patterns in color versus grayscale versions of the same stimuli. And upon doing so, he managed to identify, well, at a reasonable resolution, considering that he was using PET rather than fMRI, an area of visual cortex consistent with the position of what he called V4 in the monkey visual cortex. So this was a huge advancement, both in terms of theory. We found an area which is identified by homology with the visual cortex of monkeys, right? An area that responds primarily to color, but also it justified and validated PET techniques and their abilities to inform the study of cognition, in this case, color perception. Okay, so this was in the early 90s. Now, if we go to the early 2000s, when fMRI already became quite widely available, then Zeki proceeded to expand upon his earlier research and study more precisely the involvement of V4 in a number of phenomena that people already knew, especially from psychology. And one of those things, for instance, was color constancy, the ability to process or to perceive an object of being the same color irrespective of changes in illumination, which objectively change the appearance of an object. So we already know from psychology that the brain, our visual system, has the ability to maintain the appearance, the color appearance of an object despite considerable differences in illumination, dim lighting, extreme lighting, lighting from different positions, and so on and so forth. So what Zeki found was that V4 played a critical role in maintaining and supporting this process of inference with regard to color constancy. Okay, he also proceeded to do something quite interesting. He wondered what happens if suddenly we look at something in a false color that we do not expect. And because this still involves color processing, he found that V4 comes online reliably. But if you look at objects in their normal everyday colors, then quite a few more areas of the brain become active, because then they are associated with meaningful names and the recall of specific events and so on and so forth. So this is just building on the idea that V4 is the center of color processing in the occipital cortex, and it goes on to dissect its functionality and its ability to support a number of known phenomena from the study of perception in psychology. So now let's look at something that should already sound familiar. We already know about V1, for instance, about primary visual cortex, that it maps contralaterally. And what I mean by that is the fact that the right side of the visual field is mapped onto the left primary visual cortex, the left hemisphere, and vice versa. What about V4, right? And what he found is that if you pay attention to something in color in the left visual field, then you're primarily activating area V4 in the right hemisphere, and vice versa. So this confirms this arrangement, this mapping of left space to right hemisphere at the level of V4. Furthermore, we already knew that things are mapped from space not only in a reversed left-right manner, but also in a reversed top-down manner. And Zeki also found consistently that color processed in the right hemisphere, for instance, is mapped in the left in different parts of the visual cortex, in different parts of V4. Hence, the type of mapping of retinotopic mapping that we see in V1 also can be extrapolated into area V4. So now, what might happen if area V4 is damaged, let's say unilaterally, only on one side of the brain, only in one hemisphere? Well, expectedly, what happens is that you exhibit, as a patient, an inability to process color information within the corresponding visual field. So let's say your damage is to the left V4, then you will exhibit this very bizarre percept of the world where the right side of the visual field is devoid of color, is grayscale essentially, while the left visual field appears intact in color. And this can be even more surprising. So, for instance, one patient that exhibited a unilateral lesion to V4, meaning only in the right hemisphere, exhibited problems with discriminating color and perceiving color only to one quadrant, only to the upper left quadrant. And that certainly makes sense if we go back and we think about some of the Zeki work, which showed not only contralateral mapping, but also the up-down mapping of color information within area V4. So in this specific patient, what happened is that only color processing within one quadrant of the visual field was impacted. And now, as a point of terminology, whenever we talk about color impairment in the presence of damage to V4, the proper term for that is cerebral achromatopsia. And the reason I want to mention that is because we should differentiate between that and the much more common term color blindness, which many of you might already be familiar with, since it is quite prevalent, especially within males. And the reason I want to mention that is because in color blindness, what happens is a deficiency or dysfunction in cone level processing. So this happens at the level of the retina. Well, cones are not doing the proper job, and then individuals, especially males, fail to distinguish between specific colors. And one of the most common types of color blindness involves the inability to discriminate between red and green. So this is color blindness in the context of retinal impairment, retinal dysfunction. And this is quite different from cerebral achromatopsia. This occurs typically because of brain injury to area V4 of either one or both hemispheres. And this happens despite the fact that the eyes are doing the job properly. cones are processing color information. They pass it to V1 just fine. It's just the fact that V4 is no longer processing that information, and as a result of that, your percept of the world will suffer. So as bizarre as that might be, you might think, okay, but how bad really is it, right? There are certainly other types of brain injuries that have a much more lasting and damaging impact on someone's well-being. Well, in reality, cerebral achromatopsia is really no joke. If you think about color, and a lot of psychologists and neuroscientists have speculated about its development and its role, it is the case that one of its main functions is telling apart different types of food, right? So one theory in particular states that color development was the result of a natural selection process that emphasized the value of being able to tell different types of fruit apart. So consistent with that theory, people who suffer from cerebral achromatopsia suffer from a loss of appetite because food no longer appears in its normal color. Hence, it stops being appetizing. And this happens if only one area is damaged unilaterally, and it is even worse if both V4s on the right and on the left are damaged. So this can create long-lasting health problems in these patients. However, it is rather uncommon that only area V4, no matter how well delimited it is in terms of anatomy, be damaged in such patients. Typically, whenever you have a stroke, many more visual areas are impacted, and the area of the damage is a lot more diffuse. Hence, if you go back to one of our previous lectures where we discuss a very interesting and informative technique involving lesion overlap, it is quite relevant here to apply it in the study of achromatopsia. So because individuals such as the ones shown on the previous slide are quite rare, it is useful to see how achromatopsia might depend on the origin and the commonality of brain injury across a whole range of patients. And what this picture shows is the percent overlap of a larger group of patients who exhibit achromatopsia. And the cool colors here, the blue in particular and the purple, show the highest percent of overlap. Now, in these individuals, other aspects of visual processing were quite fine. So, for instance, face perception was relatively unimpaired. It was color processing as related to one visual field, right, the contralateral visual field, that was suffering. And it's very good to establish this kind of dissociation, right? One, as a result of one type of injury, one visual ability is preserved, but another one is impaired. So can we look at the converse of that? And some of you might already know that there is a condition called prosopagnosia or face blindness, which involves an inability to distinguish between faces. There's a lot of work on that. It can be acquired as a result of brain damage or illness. So what happens if we look at such individuals who suffer from prosopagnosia? Well, again, if we apply the lesion overlap technique, what we find is that the origin or rather the commonality in terms of lesion is quite different for these individuals. Compared to the much more localized V4 localization of injury in patients with achromatopsia. And if we want to be slightly more precise, it is the case that in individuals with prosopagnosia, the site of the lesion is a lot of the time posterior to V4, but in some cases also anterior to it. So again, this establishes the role of different cortical areas to do different things. You damage V4, you exhibit achromatopsia, but face perception is okay. You damage areas either posterior or anterior to V4, and suddenly face perception is impaired, but color perception is mostly fine. Moving on, Zeki is also known for his work with area V5, which interestingly enough is dedicated to motion processing. So following the same logic, first by utilizing neurophysiology techniques in monkeys, he found that a specific area of the occipital cortex is systematically related to motion processing. So then in the early 90s, with the advent of neuroimaging techniques, first PET and then fMRI, he was curious to see whether V5 can also be established in the human visual cortex. And for starters, he used very simple stimuli. He used flickering images of black and white patterns. He stayed away from color and even from grayscale to avoid the involvement of V4. And he contrasted those simple patterns, moving patterns, with similar series of static images. And as a result of that, he found an area anterior to V4 that was reliably and robustly activated to the first condition, but not to the second. So again, using the subtraction technique, he was able to delimit, as far as PET could allow, a part of the visual cortex that was that appeared to be specifically sensitive to movement, to moving stimuli. So again, he tried to do this with a series of other stimuli, such as checkerboards, but interestingly enough, he found that area V5 is interested even in the processing of illusory movement. And I think that many of you are probably quite sensitive to these kind of demonstrations. So I think that many of us, though not all, may look at specifically designed static images that elicit the illusion of movement. So for instance, for me, looking at this image, there is an apparent movement here, though I know, since I helped design the slide, that this is a static image. There's no animation on this current slide right now. Okay, so then the question is, does this illusion also involve the activation of V5? And indeed it does. If you apply the subtraction method to percepts that elicit illusory movement in individuals who are capable of experiencing that illusion versus percepts that do not, so the same stimuli but no percept of motion, then area V5 seems to pop up yet again. Okay, so not only is V5 involved in actual motion, but also in illusory motion. And I think there's quite a fundamental question that we should not avoid. Why do we need an area which is specifically dedicated to the movement? So for color, people have struggled to come up with an explanation, right? The importance of an area that supports color processing for the purpose of, let's say, distinguishing different types of food, or aiding object recognition in some circumstances. What about movement processing? Why do we need a dedicated neural resource to it? And here the answer is much simpler. The ability to process motion is informative not just for your ability to judge the speed and the direction of movement, but in that way it is also important for your very survival, whether that for example, serves the purpose of judging the direction and of speed of cars when crossing the street, or of avoiding an object which is thrown or is flying in your direction. So V5 supporting movement perception is critical for our very survival. Now, again, whatever was found using PET was also found using fMRI. So here is an example of area V5 overlaid on a structural MRI scan, and you can see how consistent and well delimited his location is. Now, to continue by asking a question that should become hopefully quite familiar by now. What happens if damage occurs to this specific region? What happens if area V5 is damaged? And the phenomenology behind that is quite interesting. It almost seems like the refresh rate of your brain has been significantly reduced. It's almost like watching a series of frames from a movie slowly enough that there's no longer a percept of a moving object or of a developing situation. So what we see is a sequence of static images which might suggest movement, might suggest a direction or a certain speed, but do not speak to that directly, convincingly, and powerfully enough. And that can occur as a result of damage to V5, though relatively rare. And it is a condition that bears the name cerebral achinotopsia. However, conveniently enough, V5 is located close enough to the skull that we can use TMS to disrupt its normal activity. And upon doing so, we can indeed elicit that kind of disturbing and bizarre phenomenological experience of frames of movement and of the environment in front of us, as opposed to a moving percept. Now, what happens if instead of disrupting activation in V5, we choose to disrupt the prestriate cortex, which is another name for V2, or striate cortex, which is another name for V1? Well, some disruption and some decrement in performance might occur when disrupting these areas, but it is nowhere as robust and as systematic and consistent as it is when the area that we damage is V5 at the time in which we have to perceive and judge the moving properties of such stimuli. So again, since V5 supports movement, it is not a surprise. This is rather a confirmation of the fact that if it is no longer doing its job properly, then our ability to take in visual information and make sense of movement will be severely impaired. To reiterate an earlier point and also to build on it, many of these lesions, whether true, whether genuine as a result of brain damage or disease, or whether temporary and induced artificially through techniques such as DMS, are quite diffuse. They're rarely well circumscribed. And this is a problem that neuropsychology has to deal with, not just when it comes to visual areas or V4 and V5. And this is just one such example of a lesion that induces severe akinetopsia. And this is consistent with damage to the V5. However, if you look at the gray area outlined here in both hemispheres, you can see how extensive the typical damage is. It spans considerably areas of the middle temporal gyrus, and it can extend all the way back to the lateral occipital gyri. So whenever severe akinetopsia occurs, additional problems might occur as well, because neighboring regions will be partially or completely damaged as well. And that's a more general point that we should try to keep in mind. Okay, so now let's try to be a bit more specific. What does neurophysiology tell us about neurons in area MT? Well, what we can do is construct a tuning curve. In other words, the first step will be to identify the receptive field of a specific neuron. And once we do that, once we identify the particular area of the visual field that this neuron is responsive to, what we can do is vary the types of stimuli, or rather parametrically vary the motion description, right? For instance, the direction in which something travels and the speed. And this is a very common technique in neurophysiology, especially for early cortical areas and for visual areas more generally, right? We know that V5 neurons are sensitive to movement, and they have some sensitivity to shape as well. But let's stick to the simplest possible shape, which would be an oriented bar that travels in one direction or another at one speed or another. Then what we can do is build a complete plot, describe that parametrical space completely, and track down the combination of parameters that optimize or maximize the firing rate of a neuron. So in this case, that neuron happens to be maximally sensitive to a speed of 64 degrees per second. And we say degrees because here we're measuring the visual angle, the area of space over which a stimulus travels within the scope of a second. So 64 degrees per second is quite fast. And also, if you look in the upper left corner, you can see the sensitivity of the neuron to the direction of travel. So in this case, that is maximized when something is traveling up, down, and right, left, right? So something which travels in this general direction at a speed of about 64 degrees per second is what maximizes the firing rate of the neuron. Essentially, you can think of that particular neuron as a feature detector, right? And that feature corresponds to a specific direction of travel and a specific speed. And this brings me to a more general point, which will also conclude this first part of the lecture. As we move from the retina through a variety of cortical regions, starting with the primary visual cortex all the way to areas of the parietal cortex and inferotemporal cortex, what is well recognized and established is a hierarchy of processing and a hierarchy of types of sensitivity to increasingly complex stimuli. So if you think about the retina, what photoreceptive cells on the retina do is answer to light. So if light falls in a particular area of the retina, then the corresponding photoreceptive cells will adjust accordingly their firing rates. But then as that light information travels all the way to V1, suddenly we see sensitivity to simple stimuli consisting, let's say, of oriented gratings, so parallel bars such as this. And they're critical for shape perception. It is this type of stimuli that confer to us the ability to see the edges of objects and ultimately the contours of the objects around us, and so on and so forth. Pretty much everything that we see is built on what V1 delivers in terms of shape processing and even color. However, all this information is then feed forwarded to areas like V2, V3, and V4. And while we talked about V4 primarily in terms of color processing, we also know that V4 has the ability to process and develop sensitivity to more complex shapes, such as this. And we move further and further on this posterior to anterior axis from V1 all the way to the inferior temporal cortex. We start to find relative selectivity for highly complex shapes and colors. For instance, we start to see selectivity for things like faces and scenes and words and so on and so forth, which also motivates this entire discussion we've had so far about the early visual processing. These early visual areas also happen to be some of the best understood areas in terms of cortical processes. Not just in the realm of vision, but overall. And that is because, first of all, historically, they've been some of the first to be studied in detail. And secondly, because they provide the building blocks for everything else that vision does. So it's going to be very difficult to understand how a patch of neurons in the inferior, within the temporal cortex develops sensitivity to faces if we do not understand how neurons acquire selectivity for things like oriented gratings and slightly more complex geometrical shapes first. The other point that I want to draw your attention to is here at the bottom of the slide, where we indicate the fact that as we move through this hierarchy of regions from more posterior to more anterior ones, we notice a change in the size of the receptive field. The receptive field becomes much bigger. V1 has relatively small receptive fields, small areas of the visual field for which it fires, within which it captures visual information. But as we move to area V4, that receptive field suddenly covers more of that visual space. And as we move even further down that sequence of cortical areas, suddenly a neuron within the inferotemporal cortex might develop sensitivity to an entire quadrant of the visual field, and so on and so forth. So what we see is a progression of steps in how we deal with visual information, and that progression is characterized by a higher complexity of shapes to which this areas and the neurons that they contain are sensitive to, as well as an increase in the size of the receptive field to which these areas show sensitivity. Okay, well, that concludes the first part of our lecture. So with that being said, we're gonna change gears and we'll have a brief conversation about synesthesia and cross-sensory processing. Okay, synesthesia. Presumably many of you have already heard about it, and that would not be a surprise because synesthesia has become quite popular, especially within the social media over the last decade and a half. It has become probably more popular with the general public than with scientists. And the reason for that is because synesthesia is not an impairment, it is not a dysfunction. It is most of the time not the result of an injury or of an accident. It is rather a idiosyncrasy of neurological origin that impacts a minority of individuals with a very interesting phenomenological outcome. What it comes down to is an automatical activation of a sensory experience as a result of engaging in a very different type of sensory experience. For instance, just to have an appreciation for this, the most common type of synesthesia is the grapheme color synesthesia, in which you automatically see different letters or digits, numbers, in specific colors. Okay, so this is quite a bewildering and striking phenomenological experience, and by that I mean a personal, subjective, perceptual experience. Synesthesia is not transient. Most of the people appear to either be born with it or develop it from a very early age. It is something that they experience for the rest of their lives, and it is surprisingly consistent. So the type of association that you have between a letter and a color will last throughout the rest of one's life. However, it exhibits quite a bit of variability across individuals. So if I exhibit synesthesia and I associate, let's say, the letter A with a red color, then somebody else might associate A with pink or with purple, and so on and so forth. So tremendous inter-individual consistency, but intra-individual consistency, but a lot of variety across different individuals. Also, synesthesia is a family as such a phenomena rather than a single one. There are many, many different types of synesthesia associated with very different types of phenomenological experience. Interestingly enough, it is more common amongst female individuals than males. It is more common amongst left-handers than right-handers. And the prevalence estimates vary considerably, anywhere between 1% to 20% of the population. And this is, again, because it hasn't received a lot of attention in its study, but also because it is not always easy to discern the mechanism for a type of experience which is self-reported. Right? We don't necessarily have direct evidence or immediate evidence for how these individuals cross different types of sensory experience. Okay, but to gain a bit more of an appreciation for what it entails, let's quickly list a few of the main types. I already mentioned grapheme color synesthesia. So this is one of the most common, if not the most common. So in this specific type of association, seeing letters as composing a word, for instance, in achromatic display. So let's say just a common grayscale display of letters on a screen. Persons exhibiting this type of synesthesia, synesthetes, associate letters with color. So they will see a rather boring grayscale display as a combination of letters of different colors. Okay, what else could happen? This is a particular association between alphanumeric characters and specific colors and hues, right? Well, another relatively common one is chromesthesia, where sounds are associated to color. And the reports of these individuals often state that in response to a particular note, a musical note, they see colors appear, move within the field of vision, and fade as the note, the sound carrying that note also fades. And this has been commonly reported amongst musicians and composers in the past, who take advantage of this multisensory experience to compose music. Okay, a very different type is spatial sequence synesthesia, where somebody can see digits and numbers in space. So you might imagine, for instance, that this is quite useful for mathematical and scientific creativity. It's almost as if you had an internal blackboard that other people might not have for the purpose of manipulating numbers. Okay, another type is auditory tactile synesthesia. And as the name suggests, this involves associating, prompting a tactile experience, a particular sensation of perception of some part of your body being touched in response to specific notes. And again, there is a consistent association here. Different notes are associated systematically with different types of tactile experience. One of the most striking forms of synesthesia is called mirror touch synesthesia, in which, in response to seeing somebody touch their own body or being touched on their body, you experience something quite similar. So let's say you see somebody scratching their left cheek, then you might experience a sensation of being scratched either on the left or on the right cheek. So again, this is quite striking. Most of the time it has a developmental origin, though it can also happen as a result of different types of impairment and accidents, including amputation, for instance. Okay, so this barely scratches the surface of what it has been reported in the past as synesthetic experiences. So the question is, how do we verify the reality of this reports, right? How do we make sure that synesthesia is not hallucinated, but it is a genuine sensory experience? And one of the strongest arguments in its favor, and one of the most immediate ones, is consistency across time. Whether you test these individuals a year later or 2 or 10, the same associations are reported. So this is a strong but not definitive argument, right? The textbook also reports the use of Stroop test induced interference to verify the presence of synesthesia. But this is a cognitive neuroscience course, so let's see what people have done to verify the neurological basis of synesthesia. In one of the first studies that compared DTI and fMRI data in synesthetes versus non-synesthetes, what was found was a higher fractional anisotropy in synesthetes versus non-synesthetes within the within the visual cortex. And because the study was looking specifically at grapheme color synesthesia, it was quite relevant the fact that areas that are presumed to support color vision, such as V4, and areas believed to support the processing of alphanumeric characters, words, numbers, had specifically higher or larger white matter fibers, as indicated by higher level of anisotropy. Okay, so this is consistent with the idea that synesthesia is the result of an idiosyncrasy in increased levels of connectivity between areas that are doing very different things, right? So you do expect such areas to be connected, but if you suddenly boost considerably more than in normal individuals those connections, then perception can suddenly trigger very different types of normally unassociated phenomena. Okay, so what does fMRI tell us about this comparison? And again, we use DTI to look at the structural connections between different cortical areas, but it is important to relay this with fMRI data that show us the brain in action, how viewing certain stimuli or experiencing something is reflected in the level of activation of different cortical areas. And what it was found is that synesthetes exhibited high levels of activation for visual stimuli, such as those involved in color processing and grapheme processing than normal controls. So higher levels of activation, for instance, covering but not limited to areas involved in color processing. And what you might expect as a result of that is that synesthetes not only exhibit this specific and bizarre cross-sensory associations, but because they have a larger part of the cortex dedicated, let's say, to color, in the case of grapheme color synesthesia, they may also be better at discriminating colors. And in many cases, the behavioral evidence provides support for this. And analogously to that, you can find, in many cases, better abilities to discriminate sounds in those who exhibit cross associations between auditory signals and let's say somatosensory signals, and better ability to discriminate tactile stimuli, whatever that is emphasized in corresponding types of synesthesia, and so on and so forth. Okay, so this is great because the neuroscientific evidence is starting not only to confirm the reality of that phenomenological experience, but also is starting to provide us with some understanding of how it could happen. And this understanding finds ground in the organization of the cerebral cortex. So we already talked about primary visual and auditory and somatosensory areas, but we do know that a lot of cortical areas have the task of integrating information across 2 or 3 or even more modalities, right? So for instance, we have audiovisual areas, right, as shown, as displayed here in blue. We have visual tactile areas, as shown here in green. We have audiovisual face voice areas, shown here in yellow, and so on and so forth. So a large proportion of the cortex has the task of integrating information, sensory information from multiple modalities. So you might imagine if the type of processing is these regions is emphasized beyond the norm, then the result might be a cross association of multiple modalities in response to a single one. So again, this runs in parallel with the idea that it is the white matter connectivity that is enhanced between different cortical regions. And it provides a complementary explanation in that sense. So cortical regions that are responsible for integrating information can also receive more dedicated resources than in normal healthy controls. Okay, so there is consistent evidence for possible explanations and for the reality of synesthesia, but having said that, there is so much more that we do not know about it. We do not know why it is more prevalent in some individuals than in others. Let's say the gender bias, this balance that I mentioned earlier. We don't know the precise genetic component. So we do know that it is more common in families, right? But it's still, work is still in progress to figure out what that genetic component is. There are also quite a number of hypotheses as to how this can happen neurally, and we've already tried to briefly summarize two of them. So plenty of work will need to be conducted in this field, both to explain what's going on and also as a more general tool and mechanism for revealing neural processing and its general mechanisms. Now, this also conveniently leads me into one other topic of discussion for today. And this is a very powerful illustration of multi-sensory integration in the presence of a conflict. So we already know that the brain devotes considerable resources to integrating information that come from very different sensory origins, right? But then what happens if one type of sensory evidence, let's say vision, tells you something which does not jibe well with what another sensory modality, let's say the auditory modality, provides for you? And this is very well illustrated in a classic effect called the McGurk effect, within which visual information is conveyed by how the lips of the speakers move, convey something, while what you hear conveys something relatively different. So before we go any further, I'm just going to play this video for you a couple of times, and what I'm gonna invite you to do is watch the video with your eyes open for the first time, and then the second time close your eyes and listen to what's being said again. Okay, so for the first time, let's listen to it with the eyes open. And this is a relatively brief video, so let's play it now.
Ba 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 ba.
Okay, and now let's try to listen to the very same video, but with the eyes closed. So when you close your eyes, you're only going to be able to pay attention and process the auditory signal. Okay, so let's give this a shot again, and here we go.
Ba 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 ba.
Now, if you want to listen to this a few more times, a link is provided below, so you can repeat that, adjusting the audio volume on your computer. But presumably what most of us experience is a discrepancy. If we keep our eyes open, we hear something, but if we close our eyes, despite the fact that the audio track is exactly the same, we hear something different. I believe in this video what's being done is that the visual information is consistent with the phonemic sequence ga ga, while the auditory signal conveys ba ba, right? And then what the brain does in the presence of such a conflict is try to put together that information. So many people appear to report da da, so using d as opposed to b or something else to make sense of that conflicting information. And there's a very good explanation for that, which relies on how we process linguistic information. So psycholinguists have a very good explanation of why this particular combination occurs, why we solve the conflict in that particular manner. For some other people, the conflict is solved in favor of vision, though, and I happen to be one of those people. I cannot hear da or ba, I hear ga when I keep my eyes open. And for a minority of people, the conflict is solved in favor of auditory perception. But by and large, most of us should be sensitive to the conflict and should have a very different auditory experience whether we keep our eyes open or closed when being exposed to such videos. So why are we showing this? First of all, this is a example of multisensory integration. It's an example of what the brain does when different types of modalities come into play to form a consistent percept, in this case an audio-visual percept. The other reason is because this exemplifies a conflict, a multi-sensory conflict, and this happens more often than you realize. A lot of the time you have to weigh different types of evidence coming from different sensory modalities and try to integrate it into something consistent and meaningful. And in most of those cases, the brain achieves that automatically. It's got its own mechanisms for integrating information and for solving conflicts. And the McGurk effect is a classical illustration of this. Last but not least, this brings me back to the original motivation for focusing on vision in this lecture, as opposed to a buffet of random information or the most basic information concerning all the different sensory modalities. And that's because vision tends to dominate. It is our predominant sense. It is the one to which the brain tends to allocate the most resources. So in many cases, when vision is in conflict with other senses, it tends to take lead, and the conflict is often solved in favor of visual evidence rather than its other sensory alternatives. Okay, so this brings us to the end of our theoretical discussion for today, covering sensory and basic visual operations within visual cortex, and it sets up a foundation for our discussion next week on object recognition. Okay, however, before we conclude, I want to continue with a follow-up of our discussion, which is more practically and skill-oriented regarding the design of experiments from last week. And as we discussed last week, several things should be considered each time you think about coming up with an experiment to evaluate the specifics and the neural basis of a cognitive ability. Suppose, for instance, that I'm a researcher interested in working memory. Why is it important to study working memory? That is the first question that you should ask yourself, and hopefully you're not going to have a hard time explaining that in informal, colloquial terms. Let's say to your friends who don't know much about neuroscience and psychology, to your parents, and so on and so forth. So you should be able to explain this quite clearly. Motivate what you do. Motivate the use of resources, time, effort, and so on and so forth. Not to mention lab resources if you're trying to give this a fair shot. Well, intuitively, working memory is quite valuable for the things we do on a routine basis. I wouldn't be able to lecture without keeping track of what I've just talked about and what I intend to talk about next. You wouldn't be able to keep track of what's being lectured. You wouldn't be able to learn without keeping in your working memory different pieces of information that you're trying to relate with each other, right? You wouldn't be able to go to the grocery store and go through your mental list of items that you need to purchase. There are plenty of studies that show how working memory can impact cognition, and intuitively that also makes sense. The better your working memory is, the better you should be at certain types of tasks, right? So I don't think that this is the main difficulty when it comes to studying working memory, motivating it. It will become a bit more difficult when you're trying to articulate a clear conceptual definition. And as a starter, you can try to come up with something a bit more formal, a bit more informed by the concepts and the ideas that you acquired in science classes or through other types of information with a certain level of authority, of scientific authority, right? So that will be just the first step. If you're trying to do due diligence, then you can probably better go through the literature. So let's say look up for a scientist or groups of scientists that have studied working memory for many years, and in the process they have also come up with a conceptual definition, right? So you can build on the advantage of extensive expertise and borrow that conceptual definition from a research group, right? Okay, so far so good. So then what's next? Well, here's where things become even more trickier. Moving from a conceptual definition, a high-level theoretical construct, into an operational definition. And this is about how you put to the test working memory, right? So here you have to become very concrete. We cannot just target any type of working memory in any kind of individual, in any kind of circumstance. So we have to zero in, we have to narrow down the precise aspect or instantiation of working memory that we'll be looking at. So one of the questions is, what kind of information will I be studying? So let's say we're looking for verbal information within the visual domain. Let's say we're looking for letter memory, right? And of course, this has practical implications as well, right? In the study of reading. So there's a good reason we should be doing this. But then the question is, okay, we're presenting letters visually, and then we'll be studying somebody's ability to remember them. Okay, how long should we present the letters for? How long should be the delay before we ask people to respond whether they've seen a letter or not? And here we're going to be very careful because if the interval is too long, then we might be delving into a very different type of cognitive ability, long-term memory, right? So we don't want to go there. However, we can vary the delay quite a lot to see if it has an impact or not on working memory. So in this case, the delay is of 7 seconds and a half. We will be presenting a sequence of stimuli, and we also have to think how we will be presenting them, in this case in the center of the screen, so involving center vision, as reading often does. We'll be presenting this for 3 seconds and a half. Then we will delay the memory probe for 7 seconds and a half, and then we have to decide on what the probe will be and how we will measure performance. So for instance, let's say that the probe is a single letter, and it's a letter that either was part of the stimulus or it was not, right? We will present this probe for 2 seconds, and then we can measure the accuracy, true or false, on that trial, and the reaction time. How long did it take the participant to yield a response, whether true or false? Another component of the experiment will be prompting participants to pay attention. So think about it. You might not pay attention to the stimulus on the screen. And it might be something as simple as blinking when a very short-lived stimulus is presented. We don't want that to happen. Hence, many experiments involve a attentional cue, such as get ready, right? So we can get a cue, a stimulus, a delay, and then a probe, right? And the participant can decide whether the stimulus has been part of, whether the probe has been part of the stimulus or not by pushing one button versus another. So let's make things as simple for a participant as we can. Okay, so this is how we can look at working memory, right? We have stimuli, we try to keep them in mind for a while, and then we investigate whether the participant retained them or did not. And this entire arrangement, this entire sequence of steps, attentional cue, stimulus, delay, probe, provides us with one trial. Okay, is a single trial sufficient to understand the entire complexity of working memory? Is it sufficient to provide an accurate estimate of how good an individual is at keeping things in mind? Well, obviously not. So many things can happen on a trial. So let's just look here. For instance, let's say that the probe is the letter X, right? Perhaps there is a bias towards the first letter in a string, right? And you're gonna remember that better. So on one trial we have to present X, on another trial we have to present Q, and on yet another trial we'll have to present the letter O, right? So we need at least 3 trials to have a complete or a more complete understanding of what's going on here. We also have to examine a whole set of potential foils, so probes that do not correspond to anything that was presented in the stimulus. Perhaps there will be a greater degree of confusion if they have to remember X, but I'm presenting Y as a foil rather than, let's say, the letter I, right? So I have to consider not just all the aspects of the stimulus, but all the possible false stimuli, foils, right? And other things can happen as well. Perhaps a participant pays more attention in a trial than the other. Perhaps some combination of love letters is more susceptible to good remembering than others, and so on and so forth. So many reasons for which a single trial is definitely not sufficient. As a matter of fact, the more trials I have and the more varied the structure in terms of what stimuli I'm presenting and what probes I'm presenting, the better the estimate I will have of how accurate somebody is and how fast in retrieving things from working memory. Okay, so I hope this is good enough of motivation for why I need many trials. What about individuals? Is a single individual sufficient to provide me with an understanding of a cognitive phenomenon? Not at all. Let's say that we're looking at healthy individuals, at the workings of memory within healthy adults. The more participants I recruit in my study, the better of a picture I will get of what is common across these individuals in the face of individual idiosyncrasies. So perhaps some of us are better at working memory than others, perhaps some of us pay more attention than others, and so on and so forth. So the larger the size of my participant sample, the better the estimates that I will be getting at the group level. And the less prone I will be to infer the characteristics of a cognitive ability based on a possibly biased or non-representative sample of individuals. Of course, and I'm not going to go into too much detail here, but I also want to make absolutely clear what my group is, right? For instance, I do expect that working memory performance will be quite different between young adults versus older adults, right? In case some individuals suffer from a visual or cognitive disorder versus others that don't, right? So I'm also trying to keep my my group as homogeneous as possible to have a very good characterization of what my participant group is. So for many of the experiments that we conduct in cognitive neuroscience to study normally functioning cognitive abilities, we target healthy adults with no unknown neurological or cognitive or perceptual disorder. We often look at the level of education, we keep track of gender, we keep track of many other factors that are known to influence such phenomena. So for instance, when we study perceptual phenomena or language, we want to make sure that all our participants are right-handed or only left-handed, because this influences to a large extent what cognitive resources are brought into play and what neural resources support them in such tasks. Okay, however, what we've got here is essentially one experimental condition. So while I had worked quite hard to come up with an operational definition which is concrete enough, this is a bit too limited, too narrow, because right now I'm studying the impact of working memory load with a single load of 3 alphanumeric characters. So if I'm really trying to get a better understanding, let's say, of working memory load on performance, on remembering, I will need at least 2 conditions. Let's say one in which I'm presenting participants with 3 letters and one in which I'm presenting participants with 6 letters. And you can see that in many respects, these conditions have been equated. Both 3 and 6 letters are represented in central vision, not peripheral vision. Both of them imply or involve the use of uppercase letters as lowercase letters. The relative spacing of the letters has been kept more or less the same. So I'm trying to equate those conditions as much as possible so that whatever difference I get in terms of accuracy or reaction time is primarily or solely due to the factor that I'm manipulating. In this case, the number of letters that have to be kept in memory. Further, the type of testing, the probe will be equated as well. So let's say I'm asking in this case whether the letter K was remembered. So probably I will have that letter tested as a probe in the second condition as well. Okay, so now these are just 2 conditions. There are many ways in which I can complicate this design a bit more. So let's say instead of 6 letters, I can also have 9 letters or 12 letters. I can manipulate the characteristics of the visual display. So for instance, I can verify working memory for lowercase versus uppercase. So in that case, I'm getting to the relationship between vision and memory. I can also verify the impact of the delay, whether it is shorter or longer, and it is key. That is key when studying working memory, and so on and so forth. So this is just providing you with the bare bones and with the logic of designing an experiment targeting working memory. And many such experiments have been conducted in the past within the field of cognitive psychology. However, this is a cognitive neuroscience class. So where does neuroscience step into the picture here? Well, think about it. I have 2 different conditions here. So by contrasting them, for instance, using the subtraction method as applied, let's say, to fMRI, I would be able to track down regions, cortical regions, which are a lot more involved or become more involved with a higher working memory load. And a lot of such studies have been conducted in the past. So again, just in the same manner in which I contrast conditions by looking at differences in accuracy or reaction time from a behavioral standpoint, I can use the measures provided by neuroimaging tools such as fMRI or a PET or even EEG if I'm trying to capture the dynamics of how working memory works to study the neural underpinnings. Okay. So with that being said, I would like to engage you in a little experiment, in a little exercise that parallels the one that we concluded our lecture with last week. And I'm going to ask you to choose one of these 2 phenomena, lie detection or managing distractions, and go through the process of first, providing a clear conceptual definition, provide a clear working operational definition, creating a task that involves this process that also benefits from a fair control condition. And last, using or thinking about how you can use the subtraction method to identify the underpinnings of the neural mechanisms supporting these two things, lie detection or managing distractions. And you might have noticed that I already skipped the motivation, because I hope that is quite obvious for everyone why we care about lie detection and managing distractions. And unsurprisingly, there's a lot of work both in psychology and in neuroscience trying to get to a good scientific understanding of these two phenomena. Okay, so I will challenge you to take a shot at this. This is not an assignment, but I think it's highly beneficial, both for the purpose of cementing some of the reasoning and the conceptual knowledge that you acquired in this course. Also to build an appreciation of how challenging sometimes it can be to move from conceptual definitions to operational definitions, and this is one of the challenges that we have to solve as experimental scientists. And last but not least, to see the benefit of studying such things. So I would also invite you to think about potential results of such experiments. What do you think will happen when you're planning these experiments, when you run the experiments and you look at the results? So good luck with this exercise. Again, it is not an assignment. It is rather just a way to practice some of the information you acquired in this class. And with that being said, I look forward to continuing our conversation about vision next week with a discussion on object recognition.